Hello everyone, I'm presenting today early emergence of signatures associated with HCV infection outcome using single cell approach in the 2026 Immcantation user group meeting. This is the work that I did as part of my PhD under the supervision of Dr. Ravina Bull and Fabio Luciani at University of New South Wales, Sydney. So a majority of my work was revolving around single cell technologies. We use single cell technologies to get the genomics data from single cells of T cells and B cells. So the advantage of single cell remains that if we have a mass of tissue, in case of bulk sequencing, the expression of each individual cells is masked, whereas in case of single cells, we can get the gene expression of each individual cell, and we get the heterogeneity that within the population and the variability of thousands of cells. And this is particularly relevant in understanding different tissues in the body, and in case of different diseases, how the immune system is responding in case of one disease versus other, and in case of studying the cancer cells, how one type of cancer cell is different from other type of cancer cells, and what are the cell types that are infiltrating the system. So for that, we wanted to study the adaptive immune response in case of hepatitis C infection, and for that we had T cells and B cells. T cells and B cells are particularly relevant in fighting the infection when initial antigen comes and invade the system. So in case of antigen, when antigen comes, it activates the dendritic cells, and dendritic cells, with the help of MHC molecules, they present the antigen to the naive T cells. That further activates the Th2 cells, and Th2 cells further helps in the activation of B cells. And when the naive B cells get activated, they move to the germinal center. In the germinal center, there's a dark zone and light zone with the expression of certain markers on their cell, on their surface. These cells gets activated and the affinity maturation and clonal expansion, as well as BCR diversification. These cell types further, when they move out to the germinal center, they either become memory B cells or they become long-lived plasma cells. So in case of memory B cells, these cells can be recalled at a later time point, and if the infection comes, they helps readily in release of antibodies and helps in the clearance of the infection. Long-lived plasma cells are similarly, but they have antibodies that are readily released, and they are not they express different markers when compared to the memory cells. So for that, we developed a method called VDC puzzle. We had heavy chain and light chain on the surface of B cell receptors, and VDC puzzle can isolate these alpha, beta, gamma, delta receptors from T cells, and heavy light chains from the B cell receptors using the single cell technologies from paired end reads. And in case of heavy chain, we have variable segment and the constant segments, we can identify the isotypes on the variable segment. There is a B genes and C genes, and in which genetic rearrangement occurs, and these DJ genes undergo genetic rearrangement to form unique receptors which are unique to these antigens, and this happens in case of T cell receptors as well. We have which has alpha, beta, gamma, delta receptors on their surface. So this is the flow of VDC puzzle 2.0, where we had BDJ genes, and these genes are aligned to the human genome or the mouse genome to identify the reads which are aligned to the which have been mapped to the genome. And these are aligned and assembled using Trinity to undergo recombination process, and then we use IgBlast to annotate these reads, and then the process undergo several rounds of iteration to get the contig, and that contig is further aligned with the concentration with the help of blast N to get the isotypes, as well as a hidden Markov model to get the membrane or the secretory region of these isotypes in case of heavy chain. And this is true for T cell receptor as well, where we get alpha, beta, gamma, delta chains. This tool is available on VDC puzzle on the Bitbucket, and this work has been published in 2018 and 2020. So these are the exons that we used for these T cell receptor alpha, beta, gamma, delta, and in case of identify of the different isotypes, CH1, CH1, H, CH2, CH3, as well as secreted in the membrane region. So we validated this particular tool on Jogurt and 117 B cells that were further classified into naive, memory, transitional, and plasmablast. So in case of Jogurt cells, we also implemented different aligners like top hat and star. Top hat is a slow aligner, but it has more accuracy. Star is faster, but it's less accuracy in case of more number of cells, and if we need data at faster rate, then we use star. Otherwise we use top hat if we need more accuracy. And we can see in case of this as well, like we had HiSat2 as well that we later on implemented. We wanted to see the accuracy of these different aligners in the different chains of B cell receptors, and we see most of the cases top hat is like the winner here, like 100%. We get the contigs of IgH, and whereas in case of light chain we have less recovery when we compare to the heavy chain. But in case of when we compare different aligners, top hat wins. But in case of the speed, star is has higher speed when we compare with the other aligners. Also, we validated different CDR3 and isotype detection and compared it with the bracer, this particular tool. And then we had different isotypes that we wanted to detect, as well as different subtypes of memory B cells, memory, naive, transition, and plasmablast. So we see like VDC puzzle is better in accuracy when we compare with the bracer. As well as we did this validation on mouse cells as well, where we had VDC puzzle, bracer, and basic, and we see like how these tools are different and like which is performing better. We see like in case of mouse cells, like VDC puzzle and bracer has same accuracy, but basic has lower accuracy. And in case of isotype detection, VDC puzzle was able to detect more isotypes in case of mouse when we compare it with the bracer. So we use this particular method to use to apply on hepatitis C infection. We had data from hepatitis C infection at primary infection, and we had around 13 subjects, like 6 from clears, 7 from chronics from a HC and HB cohort in prison and community. And then we had this data coming from Andrew Lloyd and Rowena Bull's lab, and we used this the PBMC samples to get the B cell receptor and a gene expression profile to identify questions such as what is the difference between their repertoire and gene expression, and how this B cell receptor and gene expression is impacting the outcome. So we see in case of hepatitis C infection, if it's an acute resolving infection, there's an infiltration of CD8 T cells, CD4 T cells, and plasma cells, and which helps in the clearance of the infection. And there's expression of CD127 on the surface of CD8, which are like activated CD8 T cells. Whereas in case of acute persistent infection, there's a viral escape, there's a PD1 expression on CD8 T cells, and there's T cell exhaustion and deletion of CD4 T cells. So all this combined like doesn't help in the clearance of the infection, and it becomes persistent after 6 months. So this is true in case of around 75% of the cases that it becomes a chronic infection, and around 25 cases it clears the infection. So very early on in the lab, it was established that there's a early emergence of neutralizing antibodies in clearance that is helping in the clearance of the virus, and there's a delay in the development of neutralizing antibody in chronics. So we wanted to understand that what is driving this, and is there differences in the B cell repertoire, or there's a transcriptomic differences. So for that, we had around 13 subjects with different genotypes, these subjects, and some of them cleared the infection, some of them developed persistent chronic infection. From them, like 25, the mean time point of their age was 25 years and 27 years. 3 clears, 3 of them were male, and 1 female, and 8 males and 7 males in case of chronics. So there's like this number of TF viruses and neutralization score. This was identified with the help of other people in the lab, like Nick Arun did the neutralizing assays, and Preston and Nasir over the years helped in the genotyping these viruses, and bioinformatics work. I was involved in the bioinformatics work. So this is the gating strategy that we used for isolating the memory B cell type. We got the single cells, and then we used CD19, CD27 positive cell types, and then we had CD10 negative cells, and then further used CD27 positive cells, and then divided this into the IgD positive, and IgD negative to get the IgG positive cell types. And further, we also wanted E2 positive cells, which is the antigen receptor on the E2 protein or the HCV surface. So these are the number of cells, B cells, that we got from the flow cytometry. There is a increase in cell numbers of B cells over time in case of clears, whereas there is a slow increase in the number of B cells in case of chronics. And healthy, there's a very low number of cells. And then after getting these cell types, these cells were isolated into 96-well plate to get to perform the SmartSeq2 technology. And then we further used BDC puzzle to isolate the heavy chain and light chain. And these are the subjects that we got, the heavy chain and light chain. And most of the cases we got around, in case of heavy chain, we got 100% in some of the patients, but average recovery of heavy chain was around 70% in case of heavy chain. And in case of light chain also, there's more recovery. There's around 80% recovery from each patient, which is a good success area for BDC puzzle. And then we wanted to study what is the difference between clears and chronics. So in case of hepatitis C, we saw a preferential use of VH169 in case of hepatitis C infection. And when we did the stats, there's no significant difference apart from VH439, which showed significant difference in their heavy chain usage. And if we see the overall use, for example, VH169 is highly used in case of hepatitis C infection, as well as VH118 as well as VH333. In case of light chain, we see higher use of kappa chain 139 and 320 as well as 144. We did not observe any clonal differences here when we did the clonal analysis, maybe due to low number of cell types. Here we had around 500 cells from all these patients. So when we look at the isotypes, most of these cells were IgG1 positive in case of chronics as well as in clears. Some of them had IgDM, which is IgD positive cells, might be naive positive cells in case of chronics. So we further wanted to see what is the difference between the CDR3 length. So if we plotted that, and we observed there is a relatively higher CDR3 length in case of both clears and chronics. If it goes beyond 20 or 22, this is very unique to hepatitis C and hepatitis C as well. Usually we do not see this long tail of CDR3 20, 25, 26, 27, which was pretty strange. So we wanted to see the significant changes between the clears and chronics. We did not observe any significant difference in the CDR3 amino acid length in case of clears and chronics, but there is a difference in the CH IgD positive naive cells and CH IgD negative length. When we look at the somatic hypermutation overall between the clears and chronics, we observed chronics had higher somatic hypermutation, whereas clears has low somatic hypermutation. And this is true in VH169 containing only antibodies as well. and this is true for light chain as well. And this might be due to the persistence of these memory B cells in the system or the circulating system of the body, and when these memory B cells are evolving, they might be staying in the germline center for a long time and going continuous somatic hypermutation. So if we further correlated this with the germline somatic hypermutation in CDR3, we found there is in case of clears, we found a negative correlation. In case of clears also, we found a negative correlation. And we divided this with greater than 19 length and less than 20 length, and we observed there's no significant difference between clears and chronics in case of when the CDR3 length is less than 20. When it's greater than 19, we saw a significant difference between clears and chronics. In case of chronics, the CDR3 length or the somatic hypermutation is higher, and in case of when the clears chronic length is more than 19, clears length is less than 19, the somatic hypermutation is low as well. So this might be true, like somatic hypermutation is impacting the length of the CDR3 region. We further correlated this with the affinity as well. We saw a positive correlation of affinity and the somatic hypermutation, and this was significant as well. We also correlated this the VH169 with the neutralizing capacity of these antibodies, and we saw a negative correlation that the neutralizing capacity of the antibodies is not linked with the somatic mutation of the antibodies. And we had this neutralizing antibodies and non-neutralizing antibodies assay, and we looked at the somatic hypermutation. We observed significant difference between non-neutralizing antibodies and non-neutralizing antibodies. We further also divided this particular antibodies, non-neutralizing and non-neutralizing antibody, in the with the germline with the somatic hypermutation CDR3, and we did not observe we observed a negative correlation here, and it's not positive correlated. So we isolated the physiochemical properties of these antibodies, such as gravy, charge, acidic, basic, and polar and aromatic residues of the CDR3 region. We observed this is non-significant aromaticity. Only in case of aromaticity we observed a significant increase in clears of the aromaticity, and this is true, we can see from here as well. In case of CDR3 region, we have presence of more tyrosine residues in the form of here, if we see, and these antibodies which show these sort of signatures have high prevalence for broad-neutralizing antibodies. And this also, like in case of CDR2 region, we see like PIXF motif, which is also true for these antibodies becoming a broad-neutralizing antibodies. And if we see here, this is the figure that is adopted from this particular paper from a different lab, and then we see like in case of CDRH2 region, we have this motif, which is a hydrophobic residue in the CDR2 and the presence of tyrosine residue, which help, or the aromatic residues, which is helping in the binding of these antibodies into the E2 protein, which is present on the surface of antigen. And if you see here, this is like the surface area. In case of heavy chain is more buried into the surface, whereas light chain is less buried into the surface, which helps in the binding of these antibodies or broad-neutralizing antibodies, such as AR3 antibodies with the antigen, and helps in the neutralization of the system. So we further had transcriptomics data from these cells, and we wanted to study like, is there transcriptomic differences. We had here 13 subjects and 2 healthy subjects from this data, and we tried to cluster them, and this is like chronics and clears and healthy data. We did not see major differences in the heterogeneity of these cell types, but we were able to isolate these cells into memory B cell 1, memory B cell 2, 3, and 4. When we did the phylogenetic analysis, or pathway analysis, we did not observe significant changes occurring in different cell types, and most of them had inflammatory signatures, which was not significant difference from different clusters. So we did this healthy, chronic, and clear. So we wanted to see what are the genes which is expressed differentially in these cell types. We observed these cell types which are mainly inflammatory genes which belong to TNF alpha and FKB pathway, and inflammatory response which is observed in chronics, which is enriched mainly in chronics and less in clears. And this is the presence of different cell types, memory B cell 1, memory B cell 2, memory B cell 3 and 4, different clusters, and then how different pathway genes are expressed in these cell types. Hallmark allograft rejection has mainly expressed in memory B cell 2. Crash signaling genes mainly expressed at low level in case of memory B cell 1, and we see interferon alpha response and TNF alpha part genes which are low expression but express mainly in cluster 1 MBC1. Also we see here interferon alpha response, coagulation pathway, crash signaling in case memory B cell 2, memory B cell 3 clusters. So apart from these expecting inflammatory response, these genes are involved in coagulation as well and collecting plaque in the arteries that further affect other functions of the body. So in conclusion, early emergence of neutralizing antibody is associated with clearance of HCV infection. We observe preferential use of VH169 gene usage. There's a higher SHM in more than 19 CDR3 length immunoglobulins in chronics due to affinity maturation and re-entry into the germinal center. We also observe pro-inflammatory signatures associated with chronic infection with low expression. With this, I'd like to thank my supervisory team, Rowena Bull and Andrew Lloyd and Fabi Luciani, for all the work conducted in this study, and all the members of my team, as well as the patients in the study group. Thank you.